Hello, everyone. My name is Sun Kang, and I'm a moderator of the symposium today. I'm a statistician at NIMH, National Institute of Mental Health, and I'd like to introduce Dr. Dong-Yoon Kim before she shares the opening remarks. Dr. Kim is a mathematical statistician at the Office of Biostatistics and Research within National Heart, Lung, and Blood Institute. And she received PhD in statistics from the University of Michigan, Ann Arbor. And before joining NIH, she held a faculty position at Virginia Tech. She's serving as the president of the Caucus for Women in Statistics and also a board member for the Korean International Statistics Society. Please join me in welcoming Dr. Kim, who will now give us her opening remarks.
Thank you very much, Dr. Kang. Nice introduction. And, good evening, everyone in the United States, and good morning to those joining us from South Korea and other part of the world. Welcome to the sixth webinar in our Bridging the Gap, Advancing Research Excellence through Equity, Diversity, and Inclusion series, hosted by Gender Innovation Science and Technology Research, GISTER, and Caucus for Women in Statistics and Data Science, CWS. My name is Dong-Yoon Kim, and I'm delighted to serve as a co-chair for this event, alongside Dr. Hae-suk Lee, Director of GISTER in South Korea. This webinar series, which began last year, continued to provide a vital platform for discussing critical topics in research. Today, we are honored to have two distinguished speakers who will share their groundbreaking work. Dr. Hae-min Lee from the University of Maryland, College Park at USA, and Professor Nam-Kyong Choi from Ewha Women's University in South Korea. We are thrilled to bring together participants from across borders to engaging in those important discussions, fostering connections that bridge nations and disciplines. Thank you all for joining us today and for supporting our ongoing effort to foster equity, diversity, and inclusion. We hope you find today's talk both engaging and thought-provoking. Thank you.
Thank you, Dr. Kim. Now I'd like to introduce our first speaker, Dr. Nam-Kyong Choi, professor at Ewha Women's University, where she focuses on research and education within the Department of Health, Convergence, and the Graduate School of Industrial Pharmaceutical Science. She earned her PhD and master's degrees in preventive medicine and pharmacoepidemiology from the College of Medicine at Seoul National University, and her bachelor's degree in pharmacy from Ewha Women's University. Dr. Choi has extensive experience in pharmacoepidemiology and drug safety, holding various positions in the field. She currently serves as the Scientific Program Committee Chair for the Korean Society for Pharmacoepidemiology and Risk Management, and as a Executive Director of the COVID-19 Vaccine Safety Research Center under the National Academy of Medicine of Korea. Additionally, she also provides advisory expertise on drug safety and regulatory science for the Ministry of Food and Drug Safety and the Korean Institute of Drug Safety and Risk Management. Dr. Choi also contributes as an associate editor for the Journal of Pharmacoepidemiology and Drug Safety. Her previous roles include serving as a visiting professor at Harvard Medical School and as a research professor at Seoul National University Hospital. Today, she will discuss why sex and gender differences matter in drug and vaccine safety. Please join me in welcoming Dr. Nam-Kyong Choi.
Thank you. Thank you for a nice introduction. And good morning and good evening, everyone. And thank you for inviting me to this Bridging the Gap webinar. And I'm honored to have the opportunity to speak today on the topic of why sex and gender differences matter in drug and vaccine safety. And before diving into the details, I'd like to take a moment to express my gratitude to GISR for organizing this excellent webinar and bringing us together. This is my introduction again. And I currently lead the EVA Pharmacoepidemiology Lab, and our research focuses on various topics in pharmacovigilance and pharmacoepidemiology, and with a particular emphasis on vaccine safety and the use of real-world data to generate the real-world evidence for research and regulatory applications. Now let me share a little about how I got into this field. I began my journey at Ewha College of Pharmacy in 1998 during the exciting time for biopharma. So the industry was really booming, and there was a great anticipation for new drug development. So in 1999, just a long time ago, Korea's first new drug, Sunpla, was approved. And Dr. Daegi Kim, who was the key person in its development, gave a lecture at our university. So I attended as a sophomore and reporter for Korea's magazine, and his speech deeply inspired me. So I dreamed of developing new drugs to help patients with incurable disease. However, during my senior year, I saw this photo. It's a baby with excessively short legs and arms. So as we know, the baby was later called thalidomide baby. I was really shocked by the fact that a drug developed for human health can cause such a great harm to people. So I started to think about how to prevent such tragedies and discovered the field of pharmacoepidemiology that evaluates safety and effectiveness of drugs in population. So when some people focuses on developing new drugs, there must also be others who study the safety of these drugs to protect the human health. So that became my mission. And now, about 25 years later, I'm pleased to be here to meet all of you here. As this slide shows, the drug development involves many stages, including preclinical testing and clinical trials. Even with this rigorous testing, why do drugs still raise safety concerns after being approved? That's because the data we gather during pre-marketing trials is often just the tip of the iceberg. So that's where pharmacovigilance comes in. So it's a crucial process for detecting, assessing, and preventing adverse effects or other drug-related problems. Sex and gender differences play a critical role in pharmacoepidemiology, influencing how drugs are prescribed, metabolized, and experienced by patients. Even so, I didn't fully understand how critical this topic was until recently. Actually, last year, I had the chance to attend the 2023 BrainLink X Lab Day, and which was focused on the theme exploring life phenomena, integrating clinical science and artificial intelligence for inclusive research. And this event really opened my eyes, and it helped me to see how much more we need to consider sex and gender differences when studying drug safety and healthcare. So since then, I've been learning more about this area, and today I'm happy to share some of the insights and lessons I've gathered with all of you. So I looked into the historical timeline of NIH actions that address sex and gender differences in clinical research. And much of this information comes from the NIH Office of Research on Women's Health. In 1977, following the thalidomide tragedy, the FDA issued guidance that excluded women of childbearing potential from participating in early phase clinical trials. So this decision was made out of concern for fetal safety. However, while it's well intended, but it had unintended consequences. It created significant knowledge gaps in understanding how drugs affect women and leaving an important population underrepresented in clinical research. And then in 1986, the NIH established a policy encouraging researchers to include women in their studies, acknowledging the gaps created by excluding women. And this policy was officially published in the NIH Guide for Grants and Contracts in 1987. However, in 1990, a Gao investigation revealed that the NIH's policy was poorly communicated and inconsistently applied with limited effort to ensure gender-specific analysis of study results. And this highlighted the need for stronger measures. So between the 1989 and 1993, the NIH shifted from recommending to legally mandating the inclusion of women in research through the NIH Revitalization Act of 1993, and which required federally funded studies to include women and minorities. In 1998, the FDA introduced the demographic rule and requiring new drug applications to include analysis of safety and efficacy data for key demographic subgroups. And these regulations required NDA to provide details on clinical trial participation and analyze the outcomes by age, sex, and race. In 2001, Gao found that 8 out of 10 drugs withdrawn from the US market since 1997 posed a greater health risk for women than men. So these findings were really surprising and highlighted the need for greater attention to sex-specific differences in drug use. So here is the list of 10 withdrawn drugs, and the further drug marked in purple in A, the greater health risks for women may have been due to higher usage among them. Here is the appetite suppressant like fenfluramine and dexfenfluramine are examples of these, and as they were more commonly used by women. On the other hand, the 4 drugs marked in red B poses greater health risks for women, even though they were widely prescribed to both women and men. So drugs like terfenadine and cisapride were withdrawn due to their association with torsadepone or TDP, potentially fatal irregular heartbeat. So this is because women naturally have longer QT intervals than men, so making them more susceptible to torsadepone. The Gao further reported in July 2001 that 1/3 of new drug applications failed to meet the FDA's 1998 regulation requiring safety and efficacy data by sex. And this report concluded that FDA inadequately addressed sex differences in drug development. And then 50 years after, Gao report, and this paper published in 2015, it takes a look at the past 20 years since 1994 in US. It's focusing on efforts to include women in clinical trials and to study sex and gender in biomedical research. Despite the federal mandates enacted 2 decades ago, requiring the inclusion of women in trials and the analysis of results by sex, progress has been slow. So as the authors of the report stated, we have not achieved sufficient progress in order to truly serve the public health. So this clearly highlights how little has changed over time, even with the policies in place, and emphasizes the need to continue the efforts to bridge this gap. Although progress in addressing these issues have been slow, the concept of the sex-based physiological and pathophysiological differences are already well established. So these differences can significantly impact both the safety and effectiveness of the same medication, and emphasizing the need to consider sex and critical factor in the drug development and safety evaluation. However, the real challenge, in my opinion, lies in applying and implementing this knowledge. So bridging the gap between the theory and actionable practice remains a significant hurdle. To understand sex-based differences in drug response, we need to examine the pharmacokinetics, how the body absorbs, distributes, and metabolize and excrete drugs, and pharmacodynamics, which refers how drugs interact with the body to produce effects. Women, for example, tend to absorb oral medications more slowly due to the lower gastric and acid secretion. And drug distribution is also tighter in women, as they have lower body water and plasma volume, but higher body fat. And also the drug excretion tends to be slower in women because of the reduced renal blood flow. The differences in liver enzyme activity, like variations in cytochrome P450, that further influence drug metabolism, often resulting in slower drug clearance in women. Pharmacodynamic differences occur when the same drug concentration produces different effects in men and women due to variations in receptor binding and drug interactions. Beyond the biology, gender plays a significant role, and social, cultural, and behavior factors influence drug safety, including medication adherence, healthcare access, and reporting adverse drug reactions. Research shows that women experience 1.5 to 1.7 times more ADRs than men. It's driven by the differences in physiology, metabolism, and hormone cycles, as well as psychosocial factors like caregiving goals and occupational exposure. Sex and gender differences in adverse drug events are evident in many studies using spontaneous reporting data. And for example, it's an analysis of Swedish pharmacovigilance data from 2005 to 2012. It's found that women reported significantly more adverse events than men in 6 out of 10 antihypertensive subgroups. And these findings emphasize the need for sex and gender-specific considerations in drug safety monitoring. And sex and gender differences are equally important in vaccine safety. So women are less likely to accept the vaccine, but tend to develop stronger immune responses once vaccinated. However, they also experience more frequent and severe adverse reactions, such as fever and pain. For example, studies using the vaccine safety data from 2005 and 2011 showed higher rates of adverse events following immunization in women compared to men. So here is the result of our research. And our research group conducted a study on adverse events associated with the COVID-19 vaccines in South Korea, focusing on gender sex differences in adverse event report using the Korea adverse event reporting system. This is not yet published data. And CARES data is a national spontaneous adverse event reporting system. It's provided data on demographic characteristics, administered vaccines, and experienced adverse events. Adverse events were coded using the MADRA classifications. And also the adverse event reports associated with the COVID-19 vaccines were submitted to CARES, and including adverse events that occurred after February 26, 2021. Each adverse event was categorized under the MADRA system organ class for both categories and prepared term for specific details. And here is the definition of the serious adverse event. And reporting rates were calculated per 100,000 vaccinations, which approximately 67 million women and 65 million men vaccinated during the study period. The women to men ratio and 90-50% confidence interval were determined by dividing the reporting rate for women by the rate for men. So subgroup analysis were conducted by age group, and reporting rates were compared between the sexes using the chi-square test. And here is the result. And a total of 887,000 COVID-19 vaccine and adverse event pair reports were analyzed, and with 280 reports from men and 606,000 from women. And most adverse events occurred in individuals ages 19 to 64, and primarily following mRNA vaccines. And this figure shows that the women reported significantly higher adverse events across 17 categories at the SOC level, while men had higher reporting rates for the cardiac disorders. Men had the higher reporting rates for cardiac disorders and serious adverse events, and such as life-threatening conditions and death. So these findings align with the previous research, indicating women's higher likelihood of reporting adverse events, while men are more prone to serious outcomes like cardiac-related events. So gender differences in adverse events can be explained by two key factors. And first, immune response. And women typically have stronger immune responses to vaccines, which can increase the likelihood of adverse events. And second, the reporting behavior. Women interact with the healthcare system more often and are generally more attentive to communicate about their health, and leading to higher adverse event reporting rates. In contrast, men's less frequent healthcare visits may result in underreporting. And these biological and social factors together influence the observed disparities. So the strength of this study includes the use of vaccination numbers as the denominator for calculating reporting rates. It's offering a comprehensive overview of gender differences. And the limitations include potential underreporting and the lack of causal influence from spontaneous reporting systems. In conclusion, the study highlights the significant gender differences in adverse event reporting related to COVID-19 vaccines in Korea, and emphasizing the need for gender-specific monitoring, communication, and future researches to address the biological and social behavior factors. And here is a summary of my talk. And first one is that sex and gender differences significantly impact the drug and vaccine safety. And number two is the historical effects have addressed the gap, but challenges remain. And third one is the biological and behavior factor drive the differences, and pharmacokinetics and pharmacodynamics and immune responses and gender-based behavior all contribute to variations in drug and vaccine safety outcome. And also COVID-19 vaccine studies findings highlights the disparities. Women report to more adverse events, while men are more prone to severe outcomes like cardiac events, underscoring the need to gender-specific monitoring. Before we conclude, I'd like to leave you the one thought. By understanding and addressing the sex and gender differences, we are not only advancing the science, but also improving lives. And these efforts are about ensuring that everyone benefits equally from the medications and vaccines we develop. So thank you once again for this opportunity to speak and share this journey with you. Thank you.